think it's just gone 7 o'clock, so we'll make a start, and anybody else that wants to log in, then Cliff will bring them in after we've started, no doubt. We're very fortunate tonight to have Professor Ed James talking to us. Ed is from Southampton University Research Centre. I think the official title is Immunotherapy Research Unit. Is that right, Ed?
Centre for Cancer Immunology.
Yeah. So I know I had a tour of it before. It was obviously before COVID. A group of us you very kindly allowed to show us around that unit at Southampton there, which was very interesting. And hopefully you're going to give us an update now, because it's 3 years since you last spoke to the PCaSO group, and I think that was at our AGM in 2018. So hopefully there's been some interesting developments in immunotherapy since then, and we can get an update from you. So without further ado, I'll hand over to you, Ed, to start your presentation.
Thank you, Roger, and thanks everyone for coming. Hopefully this will work, but as with all technology, we shall wait and see. Hopefully everybody now should see a lovely screen with my presentation, and then I will now let me just put my pointer options up. So so the last time I gave a talk to the group was, seems not that long ago, but obviously it was 3 years ago, which in the current space and time, it's difficult to know how long these things are. But so what I thought I would do, and what Roger suggested, was to give you all sort of a brief overview again, a sort of catch up about what we mean by immunotherapy, and then sort of moving on to the, shall we say, the nitty gritty, i.e. what is it that we've done in those last 3 years, or where are we at in terms of new potential therapies for prostate cancer in particular. And so the title is obviously Updates in Immunotherapy and where we are now. So
could you get that on full screen at all, Ed? Can you get a full screen on that?
So it should be full screen. Is it not full screen?
Not on mine. Has anyone got full screen? Yeah.
We've got the notes view, actually.
Oh, hold on a second. Let me just quit out of that. Let me go again. Let me change that to that. Can you see that now? Is that better?
That's it. Yeah, good.
Good. Get there in the end. Okay, so just to orientate everybody, just to get their appreciation again of where cancer originates and how it comes about. Obviously, most of you probably already know, but a cancer is a consequence of normal cells accruing a number of different mutations over a period of time. And for most people, that will take many, many years, but obviously for other potentially less fortunate people, that will occur very early in life. But these accumulations of faults in the DNA occur through many different processes, those of which we do know a lot about to an extent, such as environmental influences. So things like pollution can cause damage to the lungs, and then start to create these faults in the DNA that start the process of normal cell transforming into a cancer cell. At the same time, we have normal cell processes. So in the same way in which we age over time, though the aging process, the process by which cells replicate and form new cells is not 100% efficient, particularly when it copies over DNA. And every so often when it copies over the DNA into a new cell from the old cell, it will transfer changes or mutations that it doesn't realize it's transferred. And of course, that could then start the process by which you change the way a normal cell behaves into becoming a cancer cell. Lots of people will probably appreciate the role inheritance plays. There's a lot of information now, a lot of data that we've identified through research that shows that there are a number of genes that we inherit from our parents, and different variations of those genes that may predispose us to particular types of cancer. So the main one that is usually well known is the BRCA1 or BRCA2 mutation, which is particularly bad in women, whereby it can cause early transformation of breast tissue into breast cancer. And usually now, thankfully, there's a genetic test for people who have a family history of early onset breast cancer that can have the genetic test to see if they have a mutation that's been inherited from the mother or father, that then they can start to do preemptive measures before the cancer either develops or before it takes hold. And then the final way in which cells can become cancer cells or changes in our DNA is through viruses. So the most prevalent virus that we know about in terms of this is the human papillomavirus. And most of us know human papillomavirus because it's the same type of virus that will cause warts on our skin. But particular variants, particular strains of the human papillomavirus can also cause, the predominant cause, sort of at least 99.5% of cervical cancer, the cause of cervical cancer in women, and is becoming more and more prevalent in head and neck cancers as well, affecting both men and women. And of course, there are other viruses that we know about, particularly responsible for more blood-related cancers, such as some of the lymphomas, where we have viruses called Epstein-Barr virus, which can also mutate and change. So when they infect the cells, they change the behavior of the cell. So they want to keep the cell alive because they want it to propagate and make more virus. And so that process then obviously changes the nature of the cell. So instead of dying when it's supposed to die, it keeps growing and facilitates the changes into what we know as cancer. However, despite all the arsenal of elements that are around us that can potentially cause us to have cancer, by and large, the vast majority of cancers occur in people over the age of 60. So, it does take a while for these cancers to occur. And interestingly, over the last 50 years or so, there's been what we try to identify is what makes a cancer a cancer. What is it that drives a normal cell to becoming a cancer cell, or what attributes are found in a cancer cell that makes them a cancer? And you can sort of see these all dotted around this circle here called hallmarks of cancer. But more recently, what we've identified is that one of the key things that is really important in the development of cancer is its ability to avoid immune destruction. So the ability for it to hide away from the immune system or disguise itself so that it isn't recognized as something that the immune system should react to and destroy. And obviously there are other aspects that we all should hopefully will appreciate, things like being able to resist cell death, sustaining proliferative signaling, i.e. keep growing. And so what we find is that the ability to avoid the immune system is just as important as these other elements in defining what it means to be a cancer. So for most cancers, there are set ways in which we treat them when they're diagnosed in the clinic. Primarily, we either have some sort of, if surgery is an option, we'll have surgery to what we call debulk the tumor, to remove as much of the cancer as possible. Sometimes that will be preceded by some dosage of radiotherapy or chemotherapy, just to try and shrink the size of the tumor mass. But most of the time surgery will occur, and then you'll also have radiotherapy and chemotherapy following that surgery. But obviously not all cancers are able to be treated through surgery. So there are other options available, which primarily is radiotherapy and chemotherapy. But one of the main aspects that have come to the fore over the last 10, 15, 20 years compared to these other options is the role of immunotherapy, the ability to harness the power of our own immune systems to identify and destroy the cancer. And the beauty of immunotherapy in most instances is that it's seen as a more natural therapy, in that you're harnessing something that's inherent within your own body, compared to something like chemotherapy, which is essentially a toxin, a poison that's hopefully going to kill the cancer before it ends up killing you. And so it's seen that if we can utilize our immune system, we know the immune system is there. Can we get the immune system geared up to identifying the cancer and killing it? And so we do know a lot over the last few years about the role of the immune system in cancer. And what we've been able to see is that in lots of people throughout their life, they will potentially have come across times in which normal cells have started the process to become cancer cells. However, very early in that process, your immune system has been able to detect them as something that's not quite right, something that isn't normal, identified it and killed them. And this is something that we will never really know or appreciate because it's happened at a very early stage of that cancer development, so that we won't have a lump, we won't have a mass, we won't have any sort of effects or side effects associated with the cancer that would lead us to have symptoms that we would then go to the doctor to find out about. And so sort of in the background, without us even thinking about it, the immune system is there going around the body, potentially identifying these precancerous cells, identifying them and destroying them. However, so in the bottom here in this picture, you can see what we have here is a tumor cell, and then we have a type of immune cell that we know is one of the most important cell types in our immune arsenal that's able to identify cells and kill them, and that's the killer T cell. And this does exactly what it says on the tin. It's essentially a type of immune cell that's only real focus in life is to identify things that are not right, cells that are not normal, identify and then kill them. So essentially it's what we term a nice healthy serial killer, because what it does is once it identifies a target and kills it, it doesn't just sit and wait and go, I've done my job. It will keep going and keep going to find all the cancer cells it can find and destroy them. And only once it's the number of cancer cells is diminished to a very low level will it start to pare back its response and go back to just going around the immune system looking for new targets. So this interaction between the killer T cell and the tumor cell is incredibly important, because what we need to do is make sure that the killer T cell is able to identify the cancer cell, the tumor cell, but at the same time, it also means that if it's interacting with a cell that it's a normal cell, that it does not start to kill normal cells. So it is able to make that decision about, should I kill this cell or not? Is it something that's normal or is it something that's not normal, i.e. a cancer cell? And so the main way in which this is achieved is through molecules on the cell surface called MHC molecules. It's an acronym for major histocompatibility complex molecules. And these are essentially flags that are on almost every cell in our body. There are only a few cell types that do not have these molecules, the main one being our red blood cells. But almost every cell in our body, therefore, has these flags on the surface. And what these flags do is display what is going on inside the cell on the outside of its cell. So potentially, if this cell, this cell called an APC or an antigen presenting cell, had been infected with a virus such as COVID, very topical obviously for the last 12-15 months, if this cell was infected with COVID, the viral protein such as the spike protein would be chopped up and put on the surface, coupled with this MHC molecule, displaying it to our immune cells. And so what this means is that at any point, any moment in time, this displays a snapshot of what is going on in the cell. So in the same way as this could be showing what's going on in terms of a viral infected cell, so showing bits of virus on the surface, if this had transformed from a normal cell to a cancer cell, it could also show up on display on the surface aspects of a cancer cell that would be different from a normal cell. So we know that in the process of a normal cell becoming a cancer cell, it starts to switch some genes on, switch some genes off. And the proteins that are made can therefore be chopped up and shown on the surface to show that this potentially is something to be looked at. And so when this occurs, we have our lovely killer T cell, which is just going around surveying the environment, looking to see if there's anything to be interested in. And the T cell will come in and engage its own receptor on the surface called a T cell receptor with these major histocompatibility complex molecules, MHC molecules, that are displaying what's going on in the cell. And so through this interaction here, if it's a tight fit and it fits in such a way that gives a close coupling, this will signal to the T cell, the killer T cell, that this is something it needs to do something about. If, however, it doesn't really sit properly or doesn't really bind very strongly, it will ignore it. So if this was a normal, happy, healthy cell, this interaction would occur in less than a second, and that decision will be made within that very short amount of time whether or not something should happen. It will dissociate and go off somewhere else. However, in this example, this cell here is a cancer cell. And so it's put on its surface here, displaying something from inside the cell that is not normal. And what this killer T cell has done, because of this interaction here, is able to identify that this is not normal. This shape, this display at the cell surface is not what it would normally expect to see. And so this causes a chain reaction in the killer T cell. And again, this occurs within, you know, less than a second. It makes that decision. It can take a number of minutes after that to get going and get activated, but the initial interaction is very, very short. And so what that means is that killer T cell becomes activated and becomes angry. And the first thing it does is it will kill the cell that it's just interacted with, because it knows that it's displaying something on the surface that's not normal. And if it's not normal, then it needs to get rid of it. And so what the killer T cell will do is interact here and essentially give it the kiss of death, releasing some toxic chemicals into the cell, destroying the cell. And what that does is essentially, if it's a cancer cell, we'll obviously start to kill cancer. If it's a virally infected cell, it would kill the virus, because the virus cannot exist outside the cell. But as I said before, the T cell, the killer T cell, is not happy with just killing one cancer cell. It will then, armed with this understanding that there's something going on in the local environment that's already activated it, it will then start trafficking off and searching for even more of these within the area. So if you can imagine, if you have, say, a lump, a tumor, a cancer mass, and your killer T cell gets in and identifies one of them as something to kill, it will keep going and killing as many as it can find. And the beauty of this is, one of the first things this killer T cell will do is actually divide and divide and divide and divide. So instead of having only a few, you'll end up with a few million within a number of days. So essentially, it's boosting this arsenal, this really potent arsenal that's identified that something is wrong, there's cancer cells around, and it needs to do something about it. And so I've got a short video next just to show this in action. So this is not sort of my wonderful, amazing, my illustrations that took me forever in PowerPoint. This is actually something that we can do in the lab to show what the killer T cells can do. So this here, on this video that I'm going to show, all these cells here are cancer cells. So particularly the first one that we're going to look at here is the yellow one, but all these other cells dotted around are cancer cells. And this cell up here is your killer T cell, the one pointing in red. So when I start the movie, if I can start it, why can I not start this? Oh, can I start it? Oh, why is it not starting? Why would it not start? Oh, that's useless. You may have to take my word for it.
And sometimes within Zoom, you have to make another configuration to be able to share a video, I'm afraid.
Oh, okay.
It's in the settings, but I don't know what they are, but I know that's the way of doing it.
Where is it?
Don't feel embarrassed, Ed. We're very much with you on this, in the same position.
So anyway, I'll try and get it working at some point afterwards, and if I can get it, I'll show it to you. But essentially, what happens is this red cell, well, the red arrowed cell up here, the killer T cell, comes in, identifies this as a cancer cell, and within a split second, although the video is speeded up, the killing occurs over a matter of seconds rather than minutes or hours. And it will come in, attack this cell. This cell will shrivel and be destroyed. It'll hang around a bit, and then it goes looking, searching for other ones. And eventually it'll go around all these cells and kill every single one of them. And so that's essentially what our killer T cells are doing in our own bodies when it has identified something that it needs to search out for and destroy. And so you can see how in that process, having these cells on board, or getting these cells working, getting them working in the right way, is, you know, such a potent weapon in our arsenal to try and fight the cancer, or try and, you know, at least remove a lot of the cancer cells from the body. However, with all these things, there's always caveats, and the problem is, as I noted, as we saw in that hallmarks of cancer. Cancers don't really sit idly by waiting for the immune system to come detect them and destroy them. What they do is there's essentially this Darwinian process sped up, you know, rather than say hundreds of thousands, millions of years to develop. This can occur over days or weeks, in that there's a battle between the immune system and the cancer cell. And so initially, when the cancer cell develops, the immune system may be able to recognize it really easily, start destroying it. But then relatively quickly, any cancer cells that through their division, when they're dividing, start to change their the genes that they express, so that they switch some things on and switch some things off. If in one of those processes they switch something off that is fundamental for your killer T cells to be able to interact correctly and see the cancer cell, then obviously the killer T cell is no longer able to see the cancer cell, and the cancer cell can escape. And the worst case scenario is that at that point, the immune system is no longer able to detect the cancer cell at all. And so essentially the cancer cell can grow and proliferate and divide with impunity, and obviously cause the devastating consequences if left unabated. And so that's what we see in a lot of people, a lot of patients, when they present at the clinic with these tumor masses, with these cancers, is that we can see that the immune system has had a go. It's just that the cancers have switched certain signals off to prevent the immune system from working in the way that we want it to work, or the way it should work. And these are some of the ways in which it can do that. And so these here at on that I've sort of represented as different colors are our MHC molecules, those flag molecules that are displaying to your killer T cells. And what can happen is that the cancer cell can switch off some of those, so it reduces the number. And the problem with this is that for our killer T cells to become activated, it has to be 100% sure that what it's seeing is something that it needs to attack and destroy, because the consequence of attacking a normal healthy cell is that we then start to attack normal healthy tissue, which then can lead to disease like autoimmune disease. So essentially exactly the same ways we see for autoimmune disease. But most of us are hardwired so that our killer T cells will not do anything unless it's 100% sure. And so one of the ways, the gateways to allow the killer T cell to go ahead and kill them is if it receives a strong enough signal by having lots of these interactions on the cell surface. So I showed it as sort of a single molecule interacting with a single molecule, but really it's thousands of molecules interacting with thousands of molecules. And so what it needs to do is have enough of those interactions to give a positive signal to say, yep, this is the cancer cell. You've really got to do something about it, activate, and then kill the cancer cell. But what the cancer cell does is actually reduce the number of those. And so now instead of saying having thousands, it may only have tens or maybe even single digits. And what that means is even though your killer T cell may be able to interact with the cancer cell and have an idea that it's not quite right, it will not receive a strong enough signal to get the go ahead to say, yep, this is exactly what I need to do. And so rather than go ahead and kill something that it shouldn't kill, it will sit back and say, look, I'm not going to do anything. I just don't want to do the wrong thing, which obviously is exactly what the cancer cell wants. Another way in which the cancer cell can evade the immune system, or particular killer T cells, is that it actually changes what is displayed at the cell surface. So in this instance, my analogy is sort of, in our bodies, if we're in the UK, then our bodies are talking in the language of English. However, what the cancer cell can do is change that language so that when the killer T cell comes in, it no longer is able to understand or recognize something that should be easily recognizable as something that's wrong, as something that's changed, but it won't be able to interact in a way that allows it to understand that. So essentially, the cancer cell can change that now it's displaying its information in French rather than English. And all that means, therefore, is that if the killer T cell comes in to interact with these flag molecules, it will not be able to understand anything. And therefore, because it can't understand anything, it won't be able to make a decision of whether it's something it needs to kill or something it shouldn't kill. And because it can't make that decision, it will always err on the side of caution and say, I'm not going to do anything. I'm just going to leave it as it is. Now, the main interaction that has sort of came to prominence over the last sort of 5-6 years, particularly, and a couple of years ago now, two scientists who discovered these interactions won the Nobel Prize for medicine, was this interaction that's a purely natural process. So if, again, we take the example of a viral infection, particularly pertinent, obviously, with the current situation, when we're infected with a virus, we stimulate our immune system. Our immune system gets going, and then over a period of time, our immune system gets on top of the virus is eventually destroyed, and we go back to normal. And what that battle that's going on is essentially being won by your killer T cells. So your killer T cells they're expanding, dividing to get enough, say, troops in the army to identify those cells that are infected with the virus and destroy them so that we destroy the virus. Now, at the height of the infection, we may have millions of these killer T cells specific for the virus. But obviously, when we get rid of the virus, we don't want these going around in our body just in case they start to recognize the wrong thing and start attacking something that isn't a virus and actually causing autoimmunity. So in the natural process, as we start to win the battle, as the immune system starts to win the battle against the virus and the number of viral particles starts to get lower and lower and lower, the number of killer T cells also starts to get lower and lower and lower in a carefully balanced way. So it's not something that can go wrong in most instances. But what we want to do is shrink the number of killer T cells so we don't get these incorrect responses later on in the infection. So one of the molecules that is important for that is, rather than giving a positive signal to the killer T cell when it interacts with the cell it's trying to kill, it will receive a negative signal. So that rather than kill the cell, say the viral infected cell, it will hold off and pull back. And one of these molecules is displayed here in this cartoon, and it's called PD-1. And this delivers, if that interacts with its receptor on the surface of the cell it's interacting with, that will deliver a negative signal to stop the killer T cell from working, essentially putting the brakes on, stopping it from working. And this is a part of the natural immune process as we resolve an infection. But what the cancer cells have done, quite cleverly, if you think about it, is that they have switched on these molecules very, very early on. So rather than later on in an infection or later on in the process, as the cancer cells have almost been destroyed and having only a few left, what happens is the cancer cell switches on very early. So when the killer T cells go into the cancer, go into the tumor to start to attack the cancer cells and kill them, it's confronted with a signal by binding to this from its PD-1 molecule with this PD-L1 molecule. It delivers this very strong negative signal and says, whoa, 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 don't do anything, don't kill me. You've just got to stop. You've just got to stop. And so this signal here overrides most of the other signals that the killer T cell will have, particularly against these flag molecules. So it will stop it in its tracks and stop it from killing the cancer cell. And so what we're able to do is try to block this interaction using the antibodies that have sort of come to prominence in the last few years. And so you can see how on the surface, it seems like we've got an amazing ability in our own natural immune system to identify, kill cancer cells in a really efficient way. The trouble, of course, is that cancer cells evolve relatively quickly because of the way most normal cells have a very tight regulation on what things they do and what things they don't do. So they're told when to do it, how to do it, and if to do it, and when to live or when to die. But what a cancer cell does is it completely that process is dysregulated, so it can start to turn things on, turn things off, and see what gives them a selective or survival advantage. And if it gives them a survival advantage, then obviously they will propagate in the way of survival of the fittest. If they have a survival advantage, they will then grow more readily and more quickly than other cancer cells that do not have that advantage. And so what can happen is early on, you get a lot of death of cancer cells by the immune system, but then a few will escape because they can't be interacted with. And then, of course, over time, they will grow and grow and grow to become more of a problem than the original cancer was in the first place. And so obviously that sort of paints quite a bad picture in the sense that we've got an immune system, but it just can't work. But because we've started to understand those processes that are going on within the cancer between our immune system and the cancer cells to either stop the killer T cell from working or to hide itself away from the immune system, we've been able to develop a number of different ways in which we can start to redress the balance in our favor. And these are three of the main areas that are being looked at, and particularly in Southampton, we have a lot of work going on in these areas. The first is antibody therapies, whereby what you try to do is attach a neon sign onto a cancer cell to make it easier for your immune system to identify them as something to attack and destroy. And this is what we call antibody therapy. And it basically uses an antibody that can bind to something that we know is only found on the cancer cells. And so by adding that, they will bind to what's on the cancer cells. And then by binding to those cancer cells, that will flag up the cancer cell to immune system. They can be targeted and destroyed in a much more efficient way than if they didn't have them available. The second method is DNA vaccination or vaccination. And exactly in the similar way to how we're all having vaccinations at the moment to COVID, we can identify aspects of the cancer that your immune system, your killer T cells or your antibodies might be recognizing, and we can put those into a vaccine that can be utilized on, hopefully, for lots of people with the same cancer, but more and more likely it's going to be on a more personalized basis. So we can be able to take somebody's cancer when it's been removed, look at what makes that cancer as the cancer it is, what's new, what's different, and we can target that, and we can try to vaccinate to boost your immune system to try and identify the cancer and help it destroy it. And then the third method is what I alluded to in the end of the previous slide, is what we call checkpoint inhibition. This is the sort of therapy that got all the headlines over the last few years. And essentially what that's doing is it's blocking those negative signals that are being given to those killer T cells by the cancer cells. And essentially we put it as an analogy to releasing the brakes. So all those signals that are coming from the cancer is telling the killer T cells to slow down, stop, stop, stop. And all we're doing is we're blocking those interactions to say, yep, you're fine, go for it, killer T cells, go and attack them, do what you're supposed to do. And so over the last, there's been a lot of progression from, over the last 5 or 6 years through our understanding of the immune system and understanding what we know about the immune system, and particularly what we understand about how the immune system is interacting with cancer and particular types of cancer. And so what I wanted to do is sort of go through some of the emerging potential therapies, at least for prostate cancer, some of which are already available, although not necessarily on the NHS, as we'll find out, and others that are sort of have made sort of relatively large headlines, but are more, at an earlier stage of their development, but hopefully would be able to come through in a relatively short amount of time. And so one of the aspects that we know about is the potential of cancer vaccines, particularly for prostate cancer. And so there is a treatment called Provenge that has been given, it is a treatment that has gone through the regulatory bodies. So it's been approved by the FDA and it's approved for use in the UK and in Europe. However, it's not available on the NHS because of the cost benefits that have been applied to it. And of course, with the NHS, we do have funding limits, but the premise of this therapy is that the patient will have blood taken, and from that blood, they'll be isolating these cells called antigen presenting cells. And there's a particular cell highlighted here in this picture in red called a dendritic cell. And it's called dendritic cell because it has these dendrites that stick out of the cell, and they're almost like probes to probe the surface of what it comes in contact with. In this picture, it's coming in contact with a viral particle. But what these dendritic cells are sentinels in our body that are sort of in and around all our tissues. And if they identify something that's not quite, they don't believe is quite right, they will first engulf it, and then they will break it down and put it onto the surface, and then they'll go off to the lymph nodes, into the lymph system where our T cells live, and display it to our killer T cells and say, look, is this something you need to do something about? Sometimes it won't be, but other times it will be. And so these are also known as the sentinel cells, because they're the ones that will go look for something and then go and report back. And so in this therapy, what happens is you purify these dendritic cells, these antigen presenting cells, from the blood, and then they're fed these antigens, this prostatic acid phosphate that we know is specific for prostate cancer, or is produced by cells that have become cancerous in the prostate. And what that does is it stimulates the dendritic cell, but also provides those particular molecules or proteins or fragments of proteins. In this case, this prostatic acid phosphate gets them to put them on the surface to display them, okay? And then these dendritic cells, with this displayed, the fragments of this protein displayed on the surface, is then re-injected back into the patient. And what this does is it sort of pre-arms the dendritic cells with these proteins, so that then when they go off to the T cell area, the killer T cells will have a much greater opportunity to identify this as something to attack and destroy. And once they become activated, they will then go off to the relevant area of the body, in this instance, obviously the prostate, and identify, or potentially if it's moved away from the prostate at that point, can therefore identify any cell that has this on the cell surface and destroy them. Now, this has benefits predominantly for people with more advanced disease, but obviously you can appreciate that because it's a very personalized approach, it is quite costly in terms of being able to I take the antigen presenting cells from the blood, feed them this prostate antigen, and then inject them back into patients. And so, you know, in America, it is available and it does have relatively good success. It's not seen as a curative treatment, but it does at least push back the development of the cancer for a number of months. What's also been looked at is use of the same protein, this prostatic acid phosphate, to take the whole protein and inject it with a stimulator. So in this instance, what we call an adjuvant. So in the same way as the vaccines that we've just been given for COVID, there's an adjuvant in that to help the immune system become activated to what we want it to become activated. So for COVID, it's the spike protein, but for this instance, it would be towards the PAP protein. And so you can combine them together in an injection, and that again gets the immune system stimulated in such a way that it will recognize this PAP as something that's foreign, something it needs to react to, something it needs to do something about. And then it will activate your killer T cells, and then they will go around the body looking for any cell that has any bits of this PAP protein on the surface, particularly obviously the prostate cancer cells. And once it identifies those, it will kill them. And again, this has shown some success, particularly in phase 2 trials, but again, it's now moving further on into phase 3 to see, you know, to push the boundaries and say exactly what does it do, how good is it making a big difference or not to patients. And then the final parts of sort of vaccines, the more classical vaccines that we have, these are what we call the peptide vaccines or viral vector based vaccines. So in a similar way to COVID, where we can use, say, the AstraZeneca Oxford vaccine, where we use a viral shield to take the cargo, which is the spike protein, the gene for the spike protein, into the body. We can do the same, but in this case, it encapsulates targets that are found in prostate cancer. This again is the prostatic acid phosphate, the PSA, which we all know quite a lot about, and obviously another antigen called PSMA, which is prostate specific membrane antigen. So we can put all these into the viral vectors and immunize and vaccinate people in the idea to stimulate an immune response to these proteins. And by stimulating immune response, that immune response would then be able to identify cancer cells that are making these proteins and obviously identify them and kill them. And so again, with all these at the moment, there's a lot of promise, but obviously we still are waiting for them to come in, apart from this Provenge, actually come into the clinic for regular use. Now, for other classical immunotherapies, we have the immune checkpoint inhibitors, which, you know, came in with a lot of fanfare, particularly when we were talking about melanoma cancers, where we saw, you know, something like a 25% success rate in terms of seeing tumor shrinkage and long-term responses. And so we've essentially built up an arsenal of these drugs, either targeted towards the PD-1 molecule, which is on the killer T cell to block this interaction, or the PD-L1 molecule to block the opposite side, to block the interaction, or to block another interaction that occurs called CTLA4. And all these do essentially is release the brakes on the killer T cell to go about its business. However, the promise that it's shown in some cancers is not observed in all cancers. And so what we've found from trials and from late phase trials is one of these drugs, this Keytruda or pembrolizumab, which targets this PD-1 molecule in prostate cancer, was able to see, you know, approximately in 5% of patients, we would see tumor shrink and in some instances completely disappear, which is, you know, phenomenal given that these were quite advanced disease when the people were treated. But what was also found was that when they looked at prostate cancer patients, was that they had a much greater chance of working if the cancer subset was found to have what we call a mismatch repair mutation. And this is, you know, relatively rare in prostate cancer, but it's one of those subsets of the cancer that can easily be identified from when the cancer's been looked at. And so the idea then is that whilst it may not have a great effect for all people with prostate cancer, there are these small populations or populations within that, within the whole cancer patients, that it might actually have a much greater opportunity and effectiveness compared to if we take the whole of the population of prostate cancer patients. And this is something we're learning a lot more about, and that classically all cancers were defined by where they were found in the body. But what we're now knowing by looking at the genetics that make up those cancers, that actually they form discrete groups. And so no longer is the thinking that one treatment should fit all potentially. You know, the idea is that we can be a little bit more clever and more precise with our treatments to say, is this a drug that could work for this cancer, or is it a drug that's going to work for another cancer? And so rather than give somebody a necessary treatment, you give them something that hopefully is going to work. Another aspect is using the monoclonal antibodies. And this is something that is not a new technique, but it's something that seems to be gaining a lot of traction in prostate cancer. And it's what we call an antibody drug conjugate. And essentially it's an antibody, which you can see here in the diagram, this sort of upside down Y-shaped molecule. And it's coupled with a toxin. And in this instance, it's an antibody that recognizes a particular molecule on the cancer cell surface called CEACAM5. And we know that prostate cancer cells, particularly the neuroendocrine prostate cancer cells, express a lot of this protein on the cell surface. They have a lot of it on the cell surface. So what this antibody is able to do is target and bind to those protein molecules on the cancer cell surface. And then it's also coupled with and linked with a chemotherapy drug. And what that means is that you can deliver the chemotherapy drug directly to where the cancer is, compared to what we traditionally have in chemotherapy, whereby it's usually IV and it goes everywhere in the body, but obviously targets those cells that are more rapidly dividing. And so the idea is this is much more precision medicine, whereby you can direct the chemotherapy to the place that it needs to be. So in theory, you can increase the dose a lot more than you would be able to do compared to if you gave it sort of systemically in an IV situation, because obviously you have a huge number of side effects as you increase the dose of chemotherapy. So the idea here is again that anytime you can identify a molecule on the surface of the cancer cell, particularly a prostate cancer cell, that is unique to that cancer cell or is only really found in that cancer, in those cell types, you can direct one of these antibodies with this sort of toxic payload to direct it directly into the place that it needs to be to identify and to destroy that cancer cell. And again, these are these things that have shown really, really strong promise in early phase trials. And again, is beginning that process, that almost arduous process of confirming the effects that are seen and to see how broad an effect it has over all the prostate cancers that it's utilized against. The next set and sort of the last aspect that I want to talk about is an aspect called CAR T cells. So these are relatively new therapy, but it has been in the news as well over the last few years, and there's been a number of documentaries about the benefit of using these CAR T cell therapies, particularly in blood cancers, leukemias, lymphomas. But what we've identified is that these CAR T cells could actually be utilized in prostate cancer. So the way the CAR T cells work, for those who are unfamiliar, is that, as I said before, we have the normal T cells. This is your killer T cell on the left-hand side. It has its T cell receptor, and it recognizes those flag molecules on the tumor cell. And that interaction defines the ability to specifically see the cancer and destroy it. But what the CAR T cell does is it takes the same T cell, but instead of using the T cell receptor, it utilizes a chimeric receptor that essentially has an antibody at the one point. So this bit here is a bit of an antibody that is directed towards a particular molecule on the cell surface, in the same way as that the previous slide had the antibody conjugate that was directed toward the CEACAM5. You would have the aspect of that antibody that recognized CEACAM5, but the rest actually is the same inside bit that's found on the T cell receptor to give the same signal, so that even though you're looking at something slightly different on the outside, you deliver the same positive signal into the T cell to tell it to kill whatever it's interacting with. And it's chimeric because it's a combination of this sort of antibody on the outside, but the T cell receptor on the inside. And all that does essentially is fool almost the T cell into believing it's interacting through its T cell receptor when actually it's interacting through this antibody chimeric receptor. And so this is really potent and powerful, because if you can identify a molecule on the surface that's specific for the cancer cell, you can direct any T cell to that has this chimeric, this CAR antigen receptor on the surface. And so the idea is that if you can engineer, and some people have started doing that, engineering this antibody so that it recognizes proteins that are found in prostate cancer, such as PSA, the PAP, the prostate specific membrane antigen, or this prostate stem cell antigen. If you can create this chimeric receptor that recognizes this, you can then direct these CAR T cells. These CAR Ts will be able to direct it to where these prostate cancer cells are, identify and destroy them. This obviously is relatively early days, because this technology is developing, almost every month, but it's really promising, and it's so potent, and we can see some really good results in the blood cancer that we believe that it would have similar results in prostate cancer. The one caveat we have to say is that you have to be incredibly careful that when you start to design what it's targeting on the cancer cell, it is only found on the cancer cell. Because these CAR T cells are so potent, if this antigen, this protein on the tumor cell, is found on any other cell in part of the body, the CAR T cells will go and destroy those cells as well. And there's been a couple of clinical trials where they thought they had got something that was specific for a cancer. This is a lymphoma cancer, but what they didn't realize is that it was also found at very low levels on some heart cells. And in one of those trials, one person actually died because the CAR T cells started to attack the heart and destroyed the heart cells, which caused obviously problems for the patient, which they died from. So it's one of those things that now, with all these things, it's all experimental. So we've learned a lot since then. But at the same time, if we know that all these different 4-antigen PSA, PAP, PSMA, or PSCA are specific for prostate tissue, then it's an easy win. We know that we can target that prostate cancer and destroy it. And the CAR T cells are so efficient at doing that process, then we can see that it would be a really encouraging therapy to use. Now, with all these things, when people have looked at some of these therapies and showed that they didn't work on their own, the next stage that we start to identify is using them in combination. So rather than using them on their own, we're starting to use them. So this can either be through combination of 2 checkpoint blockade therapies targeting 2 of those those inhibitory molecules, so to give a stronger positive signal, to a stronger block of the inhibitory signal, or in a slightly different pathway is to combine the immunotherapies with other therapies such as chemotherapy and radiotherapy. And the reason for doing this is because you wouldn't necessarily use chemotherapy at the dosage that you would normally use, because chemotherapy will destroy your immune cells as well. But if you do it in a way that destroys some cells, the way it destroys those cells actually stimulates the immune system. And so if you can combine the chemotherapy then with an immunotherapy that's stimulating your T cells, stimulating the immune response, you could get a double whammy, whereby you're getting the T cells in the right place at the right time to identify and get stimulated by the dead cells from the chemotherapy, and then start to attack whatever the chemotherapy hasn't managed to destroy. And in a similar way for radiotherapy will also destroy cells, and it destroys it in an immunogenic way. And so therefore you can use the same sort of process. And so those are the things that we're currently investigating. And the idea being that perhaps, with all these things, with the best will in the world, it's not going to be a one shot. We're going to have to develop, clever ways of doing this, but we have hopefully identified some of the ways through our knowledge of how things work, of being able to combine the right things at the right time to get the response we want. And so I just wanted to sort of finish this sort of end section by looking, this is something from, a couple of years ago, but these are the number of trials that are currently ongoing for prostate cancer immunotherapies. And as you can see, obviously, there's a huge number of, only makes up a small number of therapies that are being looked at in terms of prostate cancer, but we can see here there's over 120, 130 already going through, with obviously they're diminishing returns because of what they find in phase 2 is not as encouraging as what they thought. But you can see how, you know, there's a huge conveyor belt of these new therapies that are being tested in the clinical trials that hopefully will see the light of day in the next few years. And when we break them up, we can see that a lot of them are working either on their own or in combination with chemotherapies and other therapies, looking at the checkpoint blockade molecules, PD-1, PD-L1, CTLA-4, all these other, the CYP-T is that Provenge immuno sort of vaccine therapy that we've been looking at. So the idea here is just to show, you know, there is a lot of hope and there is a lot of potential light at the end of the tunnel, because these trials are continuing to go on. And this is only for prostate cancer. You know, if we take all the other cancers that we're currently, you know, trials, then it goes into thousands. And so, because the idea is that you might discover a therapy that works for some cancers that is also applicable to prostate cancer. But once it's been regulated and approved for one cancer, it doesn't take half as much time to get it approved to be used in other cancers. And so hopefully this is a little bit of encouragement that's showing that, you know, there is a lot going on, particularly in the prostate cancer space for clinical trials. And this was only a couple of years ago. So even now, this is probably, you know, gone another 25%, if not more, based on from what I've been reading. And so where do we want to end up with? And so I sort of showed this the last time I gave a presentation, but it's the concept of understanding that each person's cancer is individual to that person. And we shouldn't necessarily expect therapies that will cure everybody or have an effect in everybody. And this is something that we now know because of the way we've been able to look at what makes people's cancers the cancer in that individual. And so on the left here, we've got two different types of cancer. And through looking at genetic sequencing, we can identify that there are potentially three different antigens, three different proteins on the surface that we can target. There's a second prostate cancer here that has three other proteins. One of them is the same, but two are different. So once we're armed with that information, we know that if we treat this patient up here, patient 1, with something that targets the white stars, something that targets sort of the orange-brown diamonds, and then something that targets the blue circles, these therapies are not all going to be applicable to this cancer. Okay, the blue circle therapy would work the same, but the other two would be treating somebody for something that's just going to have no effect whatsoever, but could have potential side effects that will stop them from having other therapies. So that means that we can design drugs or develop drugs that specifically target these other molecules so that patient 2 will get a specific drug that targets the orange hexagons, a drug that targets the red rods, and also use the same drug as patient 1 did for the blue circles. So the idea is to create this arsenal of drugs that are specific for different molecules that we know can be found in prostate cancer, but not found in necessarily everybody's prostate cancer. And so therefore, you know, patient 1 comes in, they get one set of treatment. Patient 2 comes in, they've got a completely different set of proteins. They will get a completely different set of drugs. Because if you think about it, you know, many moons ago now, oncologists knew that certain therapies would only work in half of the patients, but we didn't know which half they worked in. So we gave it to everybody. But now we're able to understand those people who it probably is likely to work in and those people who will have no effect whatsoever. So rather than giving them something that they shouldn't have, then we can give them something that actually should work in them rather than waste their time and energy and effort on a drug that's not going to work. And so there you go, there's a nice animation of them killing the cancer cells. And so what does this mean then for treatment of prostate cancer? So it means obviously the goal of everything is to increase survival. So the gold standard is obviously the word cure. Now that's a difficult word because cure is difficult to appreciate, because you'd have to therefore follow somebody for many, many years to show that it didn't come back and show that if they did die of old age at some point, it wasn't because of the prostate cancer or because of whatever cancer they had. So the main thing we want to do is increase survival. So what we're sort of looking at in the short term is that we start to treat cancer as a chronic condition, that you treat it, it goes away for a bit, it comes back, we identify what it looks like, we treat again with something else, it goes away, it comes back, we treat again, and it becomes sort of a chronic process in the same way as we see for other diseases. Eventually, though, we will get to the point at which we'll be able to hopefully make people completely cancer-free for the rest of their lives. But at the moment, it's difficult to say that definitively based on what we know. And at the same time, we're developing better therapy with fewer side effects. So one of the problems that we have with lots of the drugs that we use, particularly chemotherapies, is that you have huge numbers of side effects, and from surgeries as well. There are a lot of side effects of the surgeries. So if you can identify something that minimizes that side effect but gives you the same degree of protection, then obviously that's something to be championed. And then obviously the last section, the last part, is just to say about personalized approach. Again, this idea that whilst cancers can arise in the same place in different people, it doesn't necessarily mean that those cancers are very similar to each other. And so armed with information about what makes that cancer a cancer in that individual allows therefore better decisions to be made about how to treat that cancer, or what treatment is going to work in those patients, to hopefully therefore make the better decisions earlier on in the therapy to get better outcomes. And so I'm just going to finish there. I've sort of realized I've spoken for quite a while. Hadn't quite intended to speak for so long, but I welcome any questions, and thank you all for listening intently. I can see some people on the videos. I shall stop sharing my screen, and hopefully we can go back to the room.
Okay, thank you very much, Ed. It's very interesting discussion there that you talk that you gave. I've got a question actually. First of all, you mentioned there's lots and lots of trials happening for immunotherapy. Is there somewhere that someone can go to a website and get a list of trials specifically in the southern region that are happening?
So one of the best websites I've always found is the Cancer Research UK website. They have, if you look, they have a web page that you can type in what cancer you have, and it will come up with trials that are available, and it will tell you where they are and where they're recruiting.
That's Cancer Research UK.
Yeah. So I'm trying to remember the link, but it's one of the best ways for people to identify trials, because there isn't usually a one-stop shop to be able to identify these things, and so it's much easier, and it'll tell you what stage those trials are at as well.
Right. You didn't mention the drug, I think I pronounced it right, olaparib.
Mm-hmm.
Is that still around? Because it did have good results, I think, on lung patients, wasn't it?
Yeah. So with a lot of these, it's those aspects of where it's getting to in the trials, and the trouble with most of these drugs is they show a lot of promise, and then when they expand to the phase 3, where it's the last hurdle before it gets hopefully gets approved and into patients, it's maybe not as good as the initial results. And I'm not sure about that drug, but I have heard it was progressing, so it seems like it's still in the pipeline. But the problem with you see that sharp fall between phase 2 and phase 3, and that's mainly because they just don't stand up when they get tested, or because a lot of these show very incremental changes or show no change at all, and there's no point in developing a drug that is not going to make any difference to people.
Yeah. Okay, if anyone's got any questions, I suggest you put your hand up and then unmute yourself. Brian, you have.
Yes, thanks, Ed, for an interesting and encouraging presentation. Lots of good things happening. It may be a simple question, but how do you measure the level of your immune system?
So one of the ways, so you can do what we call in a standard blood count that you can look at. You can identify the number of different cells that you have in the blood, and that can give you usually a very good indication of your state of health for the immune system. So we know the normal range of people, and that obviously age related as well. And so we can usually identify when people have an infection because it's elevated. And so we can identify those usually from blood tests very easily.
What, do you ask your GP or your oncologist for that?
So so you can ask, usually the blood cancer you can get your GP is very easy to get done usually.
The reason I ask that question is I sing in the choir. And of course, the choir is at the one end of the spectrum, is like to be the last people to be able to go back to normal because of the risk factor. And because I've got my cancer problems, if I knew that there was, I don't know if you use high or low, but if my immune system wasn't good, say, and I'm going to expose myself to that, because going back to singing, I could make a decision outweigh the two arguments and say, well, no, my immune system is not good. I don't think I should expose myself to singing.
Yeah, no, exactly. And I think the difficulty can be is that with all these things, there is always a caveat. There's always something, and usually, the counts can tell you one aspect, but it doesn't give you the complete picture, because just because you've got lots of them doesn't necessarily mean they're going to work in the right way. And that's the difficult part of knowing. Until you are confronted with the infection, you never know exactly how well you're going to do. And that's the really, really difficult position that so many people have been in and are still in. When everybody's looking to try and go back to some level of normality, is that of knowing that your immune response, your immune system is able to do what we know it could do or what it should do. But if you have been vaccinated, then there's a good chance it will be good enough. And with all these, we've got to remember that when we're vaccinated, particularly for those maybe who may have a weakened immune system or not such a strong immune system, it may not be that you're completely protected from it, but it can mean that you're protected from getting the severe symptoms. So it might mean that you might have a few days where you're not feeling great. Nobody wants that, but it's much better than being in intensive care, and fighting for your life. And then for younger people, it means, slightly different end of the scale, in that rather than feeling ill for a few days, they don't feel it at all. But obviously what we're seeing in the encouragement we're seeing from people with weakened immune systems, the data slowly coming through from those individuals, is that particularly for the COVID vaccination, is that there are some encouragements. Typically, though, you need both vaccines. You've had to have both doses to get the maximal effect. And what we're seeing is that whilst you may not be completely protected, there's a huge decrease in your risk or potential of being hospitalized, which is something which I think is really important to know, because whilst we don't want to be ill, it's if we can protect ourselves in any way, it's better than nothing.
Okay, thank you, Ed.
Okay, anyone else with a question? Yep, Lance.
Thanks, Ed. It's really interesting, and yes, again, positive, and to know there's so many trials going on at any one time. Some of those will come out really useful, let's hope. But my question is just that, will immunotherapy have good effects against metastatic cancer, or is it just really focused on the primary problem?
So essentially, there's no real barrier for it not to work in metastatic cancer at all. So the beauty of the immune system, and obviously I'm in the thick of it, my passion is the immune system, so I would not naturally say it's the best thing in the world, but the beauty is it doesn't care where it sees these things. If it will go everywhere in the body, essentially. So if it finds it, whether that's in the bone, whether that's in the liver, whether that's in the prostate, whether that's in the brain, wherever it is, it will find it and destroy it. And so the beauty is, if you can identify, the one difficulty is, I suppose, is that when the cancer metastasizes, it can slightly change to what it looked like at the primary site. However, if you can identify something that is found in the tumors, metastatic cancers, then there's no reason why the therapy shouldn't work on those as well as it would work in the primary tumor.
Thank you.
Okay, Andrew, you had your hand up. Yep. You need to unmute yourself.
Yes, I think I'm unmuted.
Yep.
Very interesting presentation, Professor James, and we are all very thankful for it to you. May I ask, actually in Southampton University, is there any research into immune therapy? Who finances it? And the flow of financing, has it been interfered with due to Brexit?
So a lot of the funding that we receive in the centre is from Cancer Research UK or Blood Cancer UK. So that they're not all, but they're a significant proportion. And of course, they're charities that have suffered obviously in the current climate. But we also get some money from the EU. So I've got an EU grant that was funded last year, which is causing some issues just because of being able to recruit people, because now people in the EU are not seen as, fellow citizens, they're seen as international people. So it just means we have a barrier because we have to go through the process of showing the Home Office and that they are valuable members of society that are going to be useful for the UK. And so, there are lots of issues that we've had from Brexit, particularly in just supply of reagents that we use, because most of these have huge depots in Europe that they just ship over to small satellites in the UK to then distribute to around the UK. And so at the very start, at the beginning of the year, we've spent a long time having to ration some of our resources to be able to deal with the backlog. Obviously some of that backlog is to do with the COVID situation, obviously, but a lot of it as well has been due to things being held up at the border. So some of the really bespoke reagents we order from a company in Denmark, and that allows us to identify some of the particular killer T cells that specifically target some cancers and allow us to understand in patients which ones patients have and which ones patients don't have. And so these are bespoke reagents that we can't get in the UK. Nobody in the UK makes them. And so we bought them in and they were held up at the border for 6 weeks because of all the silly, all the bureaucratic, nonsense that we have with papers, etc. But it just meant that when we received them, they were useless. We couldn't use them. They've been sat 6 weeks. They're supposed to be frozen, brought in, kept frozen and then used. But of course they were useless. So thankfully the company shipped us out some more free of charge. But, it shifted our research then because we had to wait for them. Things have been moved back, and so we've had a bit of a double whammy really, with everything that's gone on. And it's, we're getting back to where we want to be. It's just taking a little bit more time than we would like.
Okay. Yeah, Andrew,
how many researchers do you have who are working on immunotherapy?
So
I assume you have a crowd of PhD students,
yeah,
and some of your own researchers.
Yeah, so everybody in the Centre for Cancer Immunology is working on immunotherapy. So that's approximately 125 people. So it's one of the only dedicated centres in the UK. And I think there's only maybe 3 or 4 in the world that specifically are involved only with looking at cancer immunotherapy or cancer immunology.
Wow.
Thank you.
Anyone else with a question? Anyone else got their hand up?
Me. Roger. Me.
Ah, Brian, yeah.
Hi there. If the one of the treatments has already been approved in the UK, but it's not available on the NHS, how much would it cost to pay privately to have the treatment, and how long would the effectiveness of the treatment work?
Nice easy questions, thanks Brian. So to a certain extent it depends obviously on the types of therapy. So for some of them, for the checkpoint blockade therapy, before they were approved for certain therapies, they were looking at tens of thousands for a single go, a single course, which is not peanuts by any stretch of the imagination. And things like the Provenge, I would imagine is up there if not more for a time. And so for a lot of these therapies, it's difficult to say exactly how long they will last, but in most instances, the average is sort of 6 months, in terms of, but at the same time, what they do is, they significantly reduce the risk of death from the cancer. But again, the trouble is it's difficult, you know, as we've tried to communicate, cancers are very different in different people, and immune systems can be quite a little different as well. So it's, with all these things, it's always, people always err on the side of caution with all, you know, and I think that's predominantly, you know, it's one of those things that the NHS have decided, or NICE have decided, doesn't necessarily justify the cost implications based on the projections of the extension of life that it would give.
Mm-hmm. I think Provenge, actually, I seem to recall a figure of 80 or 90,000.
Yeah, I wouldn't be surprised.
For a course of treatment of Provenge, something like that.
Yeah, it's because it's so bespoke, because you have to, you know, purify the dendritic cells and then grow them up and stimulate and put them back in and then monitor. These therapies are never cheap. The trouble is, because they're at the cutting edge, that's the reason why.
Thank you.
Anyone else? Trawl through 2 screens we got here.
Hi, can I ask a question, please?
Alan.
Yeah. Is it because of the individualness of the cancer why it appears that there's not so much emphasis placed on a vaccine to prevent prostate cancer as there is on the work on treatment of advanced prostate cancer?
Yeah, to a certain extent. I think the difficulty with a vaccine sometimes is knowing definitively what you want to target and what is something that you can target, and sometimes as well it's making the problem sometimes actually is not that you don't have the immune response started to the cancer, it's the cancer then modulating that response once it gets going. So it's the idea that, you know, because of maybe the location of the cancer, a vaccine, say in earlier life, may not work so well because it won't necessarily get to those tissues as efficiently. And so the one vaccine we know works really well at the moment is the HPV vaccine, but that's a very different prospect because it's a virally induced cancer, and so we can target the virus very easily because you have to go through a process of infection, and then the infection then targets. But for prostate cancer, because it's a change in the natural processes to become a cancer cell, it can be quite difficult, particularly if the cancer starts to make these regulatory mechanisms that we know of and we know a lot about. If they start inducing those very early, then it can be very challenging for any immune response that you've induced through a normal vaccination to get on top of it. So sometimes numbers just is not enough, shall we say. So for vaccination, the best thing is it just gives you lots of numbers and gives you a memory to that. And so because the way the cancer is set up, it can actually block killer T cells getting into the actual tumor material. So you need another type of therapy to unblock that, shall we say.
It just seems always that the only, or one of the main inheritances I'm going to leave for my 2 sons is prostate cancer.
Yeah, and it's one of those things that, you know, with all these things, we know that prevention is much better than cure. You know, it's always about diagnosing cancer early, but if you can identify something that stops it in its tracks beforehand, then you never have to worry. It's never an issue. And it's the research is out there. It's just, you know, getting it to a point where when we immunize, we guarantee that in people it is effective, because, you know, with vaccinations, if they're not effective in lots of different people, it's just not going to work. And it could, you know, what we've identified in some people, depending on the flags that they have, makes them more or less effective sometimes at actually inducing the immune responses from vaccines. So it's getting around some of those barriers, which we know more and more about, don't get me wrong. It's just getting the research to back up our ideas and hypotheses that convinces people that it's working.
Thank you very much.
Okay, thanks. Anybody else? I think that we might have exhausted the questions now. I can't see anyone on that screen or the other screen. So, right, thank you very much, Ed, for a very interesting talk. I hope everyone's enjoyed that. We have recorded it, so yep. See everybody's
Thank you.
so hopefully it'll be going on our website, the recording, if people want to go over that again. Next month, I don't think we've got a Zoom meeting planned, but we could very well have a meeting for PCaSO members only in July, because it's our PCaSO AGM. So, and that's going to probably, we haven't got a date for that yet, but it'll be the end part of July. But we are going to hold it on Zoom, and we'll be advertising that a lot nearer the time. So I'll let everybody go. Thank you very much again, Ed, for taking the time out. If I had a bottle of wine, I'd give it to you, but it might be a bit difficult to pass it through the screen.
I'm not sure computers like wine, do they?
But anyway,
thanks for everyone.
thank you very much.
Thanks.
Thank you.
Bye, everybody.